Hello everyone, myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Jolly Grant, Dehradun. I am again with you with another subtopic on endophytes, that is endophytic entry into the host plant. Here we will discuss how an endophytic microorganisms or how microorganisms become endophyte or how they make entry inside the host plant. So, the entry and colonization of endophytic microorganisms in host plant is carried out by host plant and microbe interaction. The successful colonization of the host plant by endophytes, it is a crucial component of advantageous plant microbe interaction. So, the plant microbe interaction process is a crucial process. So, here the generally observed that reported that the host plant, they recruit their choice of microorganisms. So, entry and colonization of endophytes into the host plant include several events that occurs within the host plant, including endophytic population entrance, their motility, then transmission from one place to another, and then once they settle inside the plant tissue and then their multiplication. So, the entry and colonization of endophytic microbes in host plant we can see here. So, the rhizospheric microflora, when they come in contact with the root exudate and they move to the root exudate, and then through this attraction root exudate, if there is a wound is there, some opening is there, through this opening they make their entry inside the host plant. So, the bacterial endophytes initially attach to the root surface. When they attach to the root surface, that portion is, as I told you before, this is known as the rhizoplane. The rhizoplane is the immediate contact zone on the root surface. The later explore the potential entry sites to access the internal plant tissue. Once the microorganisms they attach themselves to the rhizoplane zone, then they explore from where they can make their entry inside the plant. So, opening in the roots where root hairs or lateral roots emerge, as well as stomata, wounds and hydatodes in the shoots are considered the main entry points for the microbe. So, the endophytes they use these gates to make their entry inside the host plant. The endophytic bacteria likely utilize these natural discontinuities in the plant body to access the internal plant tissues. Moreover, some bacterial endophytes may modify the plant cell wall by secreting cell wall cellulitic enzymes. The common enzymes are cellulases, xylanases, pectinases, endoglucanases, which facilitate bacterial entry and spread the bacteria or the microorganism inside the host plant tissues. So, one study supported this hypothesis by observing that the frequency of entry of an endoglucanase mutant of Azoragus into rice roots were decreased as compared to the wild type strain. The mutant was unable to spread to the aerial plant strain. Many colonization studies suggested that the natural cracks mean any wound, or when the lateral roots emerge, there could be any crack there. So, these are the common entry sites for the endophytic bacteria. Furthermore, some bacteria use root apex and root hairs as entry points, followed by endophytic colonization in the root cortex and vascular tissues. Now, this picture shows the points of microbial entry. When the root hair they emerge, the portion of cells become very weak, sometimes they breaks down. When they break down, then the microbes they can make their entry. And when the root hair they emerge, when they move in the soil, because of the abrasions of the soil particles, their surface is scratched. So, when surface is stretched, so they makes an opening. Through this opening, the microorganism they make an entry. The right hand side picture clearly shows that these fine root hairs, when they are emerging, these are the main points of entry of the endophyte. The bacterial niche inside the host plant. So, bacterial endophytes mostly they occupy intercellular spaces in the plant. The most likely because these areas have an abundance of carbohydrates, nutrients in the form of amino acids and other nutrients, and they likely exclusively colonize intracellular spaces of various plant parts including roots, leaves, stems, etc., even seeds also. So, the colonization can be localized at a tissue level or systematically throughout the plant body. Once the endophytes they make their entry inside the plant through root, then through root they move throughout the plant. It does not mean they will stay in the root portion only. They can move elsewhere in the plant. In the early stages of endophytic colonization, endophytes are the first observed in root hairs and subsequently in the root cortex. So, in one study, inoculated Burkholderia strain, it was observed in cortical cells, endodermis and xylem vessels. So, colonization was especially strong at primary and secondary roots and at the base of lateral roots and root tips. From there, the endophytes, they move into different parts of the plants. So, interestingly, in one study, or we can say both intracellular and intercellular colonization was observed by endophytic bacteria. So, in maize plant, another study, bacterial endophytic colonization was stronger in lower stem as compared to the stem close to the shoot apex. It means when these are the roots, so the bacteria, they have make their entry. This is the, we can say, the lower part of the stem. So, mid entry. So, the bacteria they disperse and they move upwards, and the maximum number of bacteria were here as compared to the above part of the plant. So, it depends upon the mobility of the microorganisms. If the bacteria is able to mobile, if the bacteria is able to mobile within the xylem or some other, we can say xylem or some other plant vascular tissue, then they will move here and there. If they are not able to move within the vascular tissues, then bacteria will remain in the lower portion of the plant. So, mobility of bacterial cell accompanied by synthesis of cellular enzymes may help the endophytes to spread to the aerial plant parts, including leaves and stems. So, in leaves, the bacterial endophytes have been observed in the intercellular spaces of mesophyll, in xylem tissues and substomatal areas. Now, once these endophytes, they move inside the plant, then obviously they will, it has been observed that they impart some benefit to the plant. So, some of the endophytes, they also benefit the plant in the form of nitrogen fixation. We understand that host plant, they require certain amount of nitrogen for their optimal growth. And demand for nitrogen in the production of rubisco and other photosynthetic enzymes may suggest that the important role for biological nitrogen fixation by bacterial endophytes in the leaves. The studies have shown that diazotrophic endophytes, diazotrophic means those microbes which fix atmospheric nitrogen, diazotrophic endophytes, Klebsiella variocola colonize the mesophyll cell of sugarcane leaves. Herbus pillarum colonize young leaves and shoots of wild rice. Herbus pillarum ceropidaceae colonize leaf vein, mesophilic cells and substomatal cavities of the rice leaves. And Serratia marcescens colonize the leaf sheaths and leaf parenchyma of rice plant. So, these all bacteria, they are present inside the plants. What they have done there? They have fixed certain amount of atmospheric nitrogen. So, one relatively new area of research that remains poorly studied is intracellular colonization of plant cells by endophytes. So, endophytes are known to typically colonize the intercellular species of plants, but several examples of intracellular colonization of plants by bacteria have been reported. These examples include presence of intracellular bacteria in shoot tip of banana, shoot meristem of Scotch pine, seedling roots of switchgrass and in micropropagated peach palm. So, this picture shows how a bacteria make their entry inside the host plant. So, these are the root hairs. The root hairs, this is the secretion of some, we can say, root exudate or photosynthates which are rich in amino acids, carbohydrate, proteins, minerals, etc. These root exudate, they have attracted the rhizospheric microorganisms, they move towards that, and then when they come in contact, they attach themselves and then they make their entry inside and the microorganism move inside. The root hair point of entry we discussed. The root hairs offer a logical point of entry for all type of endophytes. The many cases of intracellular plant microbe interactions begin with colonization of the microbe through intracellular access to the root hairs. So, we understand that, we have read also that well established legume rhizobum symbiosis and is one method reported to be used by some endophytes. So, endophytes generally follow those methods which have been followed by other rhizospheric bacteria or rhizobum bacteria. So, endophytes may be capable of gaining access to the intracellular space directly by secreting cell wall degrading enzymes and or through a phenomenon known as rhizophagy. Now what is rhizophagy? It's a very interesting phenomenon. Let's understand this. Rhizophagy is a cycle which refers to a plant's ability to farm or cultivate the beneficial microbes they need. So once they cultivate these microbes, then plant extract this farming process through the attraction of beneficial microbes to the rhizosphere by producing the exudates they feed on. So first picture shows the microbes exit zone, we will discuss later, and there is the A. So plant cell entry zone, microbe through these they make their entry, and then B, the plant they make the microbes they goes inside, and then microbe goes inside. Once they goes inside, the nutrients captured by the microorganisms, they are extracted by the plant roots. So in a process, the team has named the rhizophagy cycle. Rhizophagy means root eating. The bacteria and fungi cycle between the free living phase in the soil and the plant dependent phase within the cells of the plant roots. The microbes, they obtain nutrients, nitrogen, phosphate, other minerals from the soil, and the nutrients are extracted from microbes in the cells of the plant root. This is the picture of Phragmites. This is giant reed grass with a cloud of bacteria around the root tip where bacteria are assembled. Where the bacteria, they will make their entry inside the plant. Now this rhizophagy cycle step involves 12 steps. So number 1, the microbes colonize the root tip meristem where plants secrete exudates. Microbes establish intercellularly within the plant root cells. Then cell walls are oxidized of microbes using superoxide produced by the root cells. It means the superoxide they are produced by the root cells, and then the constant movement of microbe protoplast around the root cells is known as cyclosis, and exposure to superoxide produced on root cell plasma membrane extracts nutrients from the microbes protoplast. Microprotoplasts are replicated within the root cells so that many clones of internalized microbes are made, and then internalized microprotoplasts stimulate root hair formation in the root epidermis. The microprotoplasts then ejected back into the soil from the pores in the tip of elongated root hairs. So the microbes, the protoplast, they stimulate the root hair formation. When the root hair formation is there, they enter into the root hair, and from the root hairs, when the root is growing, they are ejected into the soil. Then root hairs secrete exudates into the recently ejected microbe from the tip of the root hairs. The microbe protoplast, again they form the cell wall. Once ejected back into the soil, microbes colonize the rhizosphere. They acquire again additional nutrients they need to grow in the rhizosphere. Then microbe recolonize the root tip meristems, restart of the rhizophagy cycle. Now rhizophagy cycle works like this, a very interesting cycle. Plant cultivate essentially farm microbes around the root tips by secreting root exudate or photosynthetes. The microbes grow and then enter the root tips at the tip where the cells are dividing like hardened walls. The microbes lose their cell walls, become trapped in the plant cells, and are hit with a reactive oxygen, that is superoxide. The reactive oxygen breaks some of the microbial cells. So when the cell is broken, the nutrients comes out. These nutrients are utilized by the plant roots, and then these microbes, they are thrown out of the cell or thrown out the roots into the soil. And then again microbes acquire the nutrients, and this process goes on. Lastly, the key points: the endophytes make an entry through root hair, stomata, broken cell wall, growing root hairs. Inside the host plant, the endophytes, they establish themselves. Some of the endophytes remain in the roots, some of the endophytes they move in the aerial part of the plant. Reported to have beneficial relationships. Once these endophytes they move inside the plant, they multiply. When they multiply, they are reported to impart positive benefits on the host plant, and these microbes they also show this interaction of plant microbial endophytes, a process of rhizophagy, and which plays an important role in providing nutrients to the host plants. In rhizophagy, the nutrients are extracted from the bacterial cell, and these nutrients are utilized by the host cell. So this was all about the today's lecture. Thank you so much.